In the mid-1800s, scientists like Louis Pasteur and Robert Koch developed the germ theory of disease, which revolutionized medicine. They proved that bacteria, tiny living organisms, could cause diseases in humans and animals. Pasteur's experiments on wine, beer, and milk showed that spoilage and illness were caused by microorganisms, not bad air or mystical forces. He also developed pasteurization, a method of killing microbes by heating liquids, which is still used today. Robert Koch provided the first experimental evidence linking bacteria to disease. How bacteria were grown in labs. When bacteria were placed on agar plates, they multiplied into visible colonies. Why some diseases didn't fit the bacterial model, and why scientists suspected something small. Certain diseases, such as tobacco mosaic disease in plants, confused researchers because no bacteria were visible under microscopes. Attempts to culture or isolate bacteria failed, yet the disease still spread from infected to healthy plants. To investigate this mystery, scientists used ceramic filters designed to trap all known bacteria. These filters had pores smaller than the size of bacteria. When liquid from infected plants was filtered, it still caused disease. This surprising result led scientists to suspect an agent smaller than bacteria. They proposed a new type of infectious particle, not visible under microscopes and unable to grow on standard media. This unknown agent was named virus, from the Latin word for poison. How special filters remove bacteria. Clarification on size. Chamberland and Brookfield filters had porous ceramic walls with pore sizes around 200 nanometers or smaller. Most bacteria measure between 200 nanometers and several micrometers. They are too large to pass through 200 nanometer filters and are therefore trapped. Most viruses range from 20 to 150 nanometers, far smaller than bacteria. Viruses like tobacco mosaic virus, TMV, were small enough to pass through filters. Bacteria were removed, but the virus remained and infection continued. This confirmed that the infectious agent was smaller than any known bacteria. At the time, no scientist had visualized a virus, leading to the belief that viruses were fluid-like and non-cellular. How scientists understood viruses require living host cells. Only caused infection when placed into a living host, such as a plant, animal, or bacterium. This showed viruses are obligate intracellular parasites. Viruses inject genetic material, DNA or RNA, into host cells. They hijack the host's cellular machinery to make more virus particles. Before the 1930s, light microscopes couldn't detect viruses due to their tiny size.
Some objects are so small that they cannot be clearly seen.
Invention of the electron microscope in the 1930s, scientists could finally see viruses for the first time. This confirmed that viruses were real, physical entities with their own distinct biology.